Good morning, everyone. I just want to thank the organizers for giving me the chance to speak about my project where we are using single cell to identify tissue-specific and circulatory immune signatures in Crohn's disease. So our lab is interested on IBD, which is a chronic, typically progressive and debilitating condition of the gastrointestinal tract. The main subtypes are Crohn's disease, characterized by patchy inflammation throughout the small and large bowel, typically occurring in the terminal ileum. And then we have ulcerative colitis, characterized by a more continuous and uniform inflammation in the large bowel. Now, the pathogenesis of IBD, it's not completely understood, but it's believed to be driven by a dysregulated immune response against commensal gut bacteria in genetically susceptible individuals. Now, one of the problems of trying to understand why this aberrant immune response happens, it's the fact that immune cells are highly dynamic, and in very broad terms, they can be categorized into two. So the first ones are resident cells that provide localized and long-term immune protection in the tissue. The second group are circulatory cells that patrol the body via the bloodstream and can be recruited to the site of inflammation to provide additional support. Now, in the last decades, we have really improved our understanding of how immune cells respond to local signals. But for chronic conditions such as IBD, it is unclear whether this ongoing inflammation results in new transcriptional changes. And in the case of circulatory cells, to what extent they have the same functional capacity and they can mimic the disease environment. This is particularly relevant for biomarker discovery and also personalized medicine because blood represents an easily accessible tissue. So to really answer these questions, we turn into our IBD Verse project, where we have collected 125 Crohn's disease patients, along with detailed clinical information. As part of this clinical information, we collected endoscopy scores that quantify the inflammation at the terminal ileum. In this case, a score of 0 represents no inflammation or very mild, whereas a score of 9 represents higher inflammation. From these patients, we collected per terminal ileum and blood biopsies, and then we performed single-cell RNA-seq sequencing, resulting in almost 900,000 immune cells across 47 immune clusters. Now, the 47 immune clusters are in the x-axis, and then the y-axis is just showing you the number of cells colored by the tissue of origin. And as you can see, our atlas spans different subsets of T cells, B cells, and myeloid cells. And I'm just going to highlight a particular example in the case of T cells, where we were able to identify a pro-inflammatory cluster of CD4 TRM cells that it's characterized by elevated expression of CD161 and CCR5. This cluster was previously characterized and has innate and effector activities, as well as being the predominant producers of type 1 inflammatory cytokine. So as you can see, our atlas is already able to find pro-inflammatory signals associated with the mucosal inflammation happening in the gastrointestinal tract. But really, our main question to doing this atlas is how well circulating cells can capture mucosal inflammation. So to do that, we perform a differential gene expression analysis, and we identify tissue-specific, but also share correlation with endoscopy scores. So to do this, we follow a pseudo-bulk approach where we collapse cells by cluster, tissue, and donor. So now in the second bar plot, you just can see the number of samples we had for each cluster. And I want to highlight that among, again, T cells, we found that at the sample level, we actually had some cells that were sampled from the blood and were clustered with these tissue-resident memory cells. Now, at the absolute number of cells, we can see that this cell is comprised mostly from the terminal ileum, which is what you would expect based on literature. But then at the sample level, we had more than 45 donors with at least 10 cells that were coming from this cluster, meaning that this is probably not donor-specific nor batch effect. These cells were clustering together with the terminal ileum TRM cells, meaning that these cells are transcriptionally more similar to their TRM counterparts than other population of T cells, either circulating or tissue specific. In terms of gene markers, they were also expressing higher levels of genes associated to TRMs compared to other cells from the blood. Similarly, they were downregulating genes associated to circulatory cells. So we hypothesized that given the large number of donors that we have with these cells, these are actually XTRMs. So these are cells that egress the tissue and re-enter the circulation. XTRMs have been previously characterized in mice, including intestinal-derived XTRMs, but human studies have only focused on skin-derived XTRMs. And I want to highlight that we do have expression of CCR9, which is the gut homing receptor, and it's specific to intestinal TRMs, meaning that these are indeed coming from the intestine and not skin or other tissues. Given the large scale of our data set, our atlas is able to provide the first evidence of circulating XTRMs in human gut and Crohn's disease patients. Now, this is actually quite important because when we perform our differential gene expression analysis and try to look for correlations with mucosal inflammation, XTRMs were the only circulating cell that had significant genes correlated with mucosal inflammation, as shown in this third bar plot. So here the bars are colored by whether genes were significant in the terminal ileum in like yellowish. They were significant in both tissues, colored by gray, or only in blood colored in purple. But as you can see, we don't have any significant gene that was correlated with mucosal inflammation in the blood. So now I'm just gonna show you a particular example of a gene called JAK-MP1. This gene regulates cytoskeletal dynamics, enabling T cells to move through tissues, locate antigens, and interact effectively with other immune cells. So as you can see in these box plots where the x-axis are the endoscopy scores and the y-axis is the gene expression, we can see that there's a positive correlation in both subsets of CD4 TRMs. Now, if we looked as well at the blood, we see a similar pattern, meaning that there's a positive correlation. And even though that in the case of the pro-inflammatory subset, the gene expression levels are lower compared to terminal ileum, we still see this same positive correlation. Now, this gene was not only significant for TRMs, but also for Tregs. So in the case of Tregs, we had two different clusters, one that was having higher expression of TRMs and another that didn't. The second cluster of Tregs are probably cells that were just recruited from the periphery to the intestine, and hence why they were initially sampled from the terminal ileum. Now, again, you can see there's a positive correlation, meaning that higher mucosal inflammation is correlated with higher expression levels. In the case of these CD4 Tregs, we also have cells from the blood. But now if we see at the gene expression levels, we don't see any significant correlation, and the levels are the same across all the scores of the endoscopy scores. So this is just a particular gene, but we actually had 98 significant genes for the CD4 TRMs, 84 for the pro-inflammatory cluster, and 3 for CD8 TRMs. So we actually wanted to be more systematic and look at what pathways are correlated with mucosal inflammation in both tissue TRMs, but also the XTRMs. So we performed a gene set enrichment analysis, and we found that both subsets of CD4 TRMs were enriched in cytokine signaling, and I'm just showing you all the genes that were associated to this pathway here. Interestingly enough, the pathways by which they were doing the cytokine signaling were actually different. So in the case of CD4 TRMs, we saw mostly an enrichment of genes associated to interferon gamma signaling, whereas the pro-inflammatory pathway had more enrichment of interleukin signaling, but also TNF. And I want to highlight that these both pathways are already targeted by different IBD meds. Aside from the CD4 TRMs, these genes were not only present in TRMs, but also on Tregs with TRM phenotype, as well as macrophages that have high expression of an integrin gene. These two cell types have key roles in the pathogenesis of IBD and are known to increase inflammation, further supporting our roles. So in summary, we created a single cell atlas of intestinal and circulating immune cells from 125 patients, encompassing almost 900,000 cells across 47 immune clusters. We provide the first evidence of gut-derived XTRMs in human gut and Crohn's disease patients, both CD8, CD4, and also the pro-inflammatory subset. XTRMs were the only circulating cells that had genes that correlated with mucosal inflammation, and the direction of effect were the same as their tissue counterparts. Key pathways that were correlated with mucosal inflammation included interferon gamma, TNF, as well as interleukin signaling. And these, again, were shared with Tregs and macrophages. And finally, potential future work. Now we are exploring XTRM as blood-based biomarkers for monitoring disease activity. So this is all. Thanks, everyone, from listening. I just want to highlight that we are hiring, and we also are always very happy to collaborate. As you can see, we have a very big data set, and we are just exploring 1% of our data. So if you are interested, come talk to me. Yeah. Happy to take any questions now. Questions?
So really nice talk, Lucia. I'm going to see the JAK1 gene to know if they are if the gene is driving inflammation or is a consequence of inflammation. Did you check the expression of this gene in other contexts to see if this is a consequence?
Well, even if you compare, for instance, across healthy individuals, you wouldn't really be able to tease whether it's a cause or a consequence, unfortunately. In our lab, we usually try to use genetics to try to couple whether this gene is driving or being just a consequence. This is something we are actively working on. We also are doing EQTLs. So there might be some insights there, but I'll have to check. And in terms of drug targets, no, there are no, as far as I'm aware, drug targets for these JAK, either for IBD or other immune-mediated diseases.